All right, hello everyone. Thanks very much, Morgan. Yeah, so today I'm going to be talking about ensuring interoperability of oncologic datasets and APIs through GA4GH standards and testbeds. So just to give a bit of an introduction, I think most people here have probably heard of the GA4GH. If you're not sure what we do, our mission is sort of to facilitate the sharing of genomic data worldwide and facilitate data sharing by making the data sets and APIs standardized and interoperable with one another. So we're essentially seeking to harmonize approaches to data access, data analysis and things like that. Typically, data can be in silos. There's different genomic domains, different countries with different regulations, different institutions with different ways of doing things. And so we seek to harmonize approaches to representing genomic data to lead to bigger and larger scale analyses. So just some figures about the GA4GH. We currently have 580 member institutions across 50 different countries. We are comprised of 24 driver projects, and so these are large-scale worldwide projects that involve lots of sequencing of many genomes. And I think the projection for the next few years is for the 24 driver projects combined, there's a goal to sequence about 3 million genomes. Our work is divided into 8 work streams, and these are where work gets done. There are calls to develop standards and to propose different ideas of different methods, different ways of accessing data. And currently we have 28 standards released. So some of the ones that you might be familiar with are the SAM, BAM, and CRAM file formats for alignments. We also have VCF and BCF for variant representation, and as well as the TERS API, the tool registry service. There's a team here that works on DocStore, which is an example of a TERS API. So what is a standard? Just to go over it briefly. Essentially, you can think of it as like a contract or an agreement that, here's one proposal for how data should be represented. And so many groups will agree to represent their data in a standardized way so that the data can be moved from one site to another and everyone will understand it. So if you look at an example like a file format, we all agree to represent our alignments as a BAM file. And so if it moves from one researcher to another, everyone can interpret and understand that data. So in terms of the GA4GH standards, we sort of have our standards basically fall into one of several different broad categories. These can be viewed as like an API specification, which essentially states different methods for accessing resources, genomic data over the web. A file format specification, so how is a series of bytes represent a genomic file, like a BAM file or a VCF file. We also have schemas or data models. This is a way to represent like phenotypic information or a clinical assessment. Can we have a standardized way of representing that data? We also have frameworks and policies for things like regulatory ethics, privacy concerns, data security concerns, and things like that. So my talk is going to be focused most on the API specification side of things. So what is an API specification? Essentially, it's a way to standardize HTTP methods for requesting certain data sets. So how an API specification is developed at our organization. So as I mentioned, we have the GA4GH work streams, which are essentially groups of volunteers from different driver projects or different genomic projects somewhere in the world. And they will, provide their input, their best practices, their best ideas for how to facilitate the access of genomic data. Once this goes through several rounds of, product development and product review and committee processes, this will get finalized as an open API specification, which lives in GA4GH space, and this can be considered like a source of truth. Once that source of truth has been officially released, then we say, please adopt this standard in whatever context, then any sort of driver project or any other implementer anywhere in the world can deploy their own implementation of that standard. And so what we're talking about here when we talk about API, we're only talking about access methods. So, get this file by this method or get this file by some other means. We're not talking about infrastructure, so we don't necessarily endorse like one set of infrastructure like AWS or Google Cloud or anything over the other. However, the implementer wants to develop their infrastructure, as long as they implement the API completely, then we say, that's a valid implementation of a GA4GH API. So just to go through some API specifications that might be more relevant to cancer research in general. So we have the RefGet standard, and so the RefGet is essentially an API for accessing reference sequences over the web. Some of the problems related to genomic research deals with ambiguous identifiers for reference sequences. So maybe a certain accession number, there might be multiple reference sequences with the same accession number, and this can lead to errors or inconsistent results downstream. So RefGet essentially seeks to eliminate this sort of ambiguity by having sequence identifiers derived from the sequence content itself. This is essentially done with like a hashing function. So if you have a reference sequence, you perform some sort of checksum digest hashing function on it to get a digest value, and then that ends up becoming the identifier. So it removes all ambiguity by having the ID directly derived from the sequence hash. And then on top of this, there's sort of a REST API. So that's the API layer to it, where you can access reference sequences, collections of reference sequences like an assembly, or metadata associated with it. Moving on, so we also have htsget, which is HTS for high throughput sequencing. So this is more for accessing alignment files, so SAM, BAM, and CRAM. If anyone uses SAM tools 1.7 or later, you might actually be using htsget without realizing it. So if a server is an htsget server and you request a BAM file via SAM tools and you specify a URL on the command line, then if the server is an htsget server, it will be built to handle these types of request response flow. So essentially with htsget, it's supposed to sort of streamline the process of accessing large-scale genomic files. If a researcher is only interested in accessing a part of the BAM file, or they're only interested in a genomic locus of interest, they can specify via the API certain genomic coordinates, and they will only receive those genomic coordinates. So the entire BAM file, if it's too large, it will need to be transferred. So it's supposed to facilitate more efficient transfers of genomic data, essentially. There's also the way that the API is structured is that it allows for downloading multiple file parts in parallel, allowing you to take advantage of distributed computing if that's sort of your infrastructure. Moving on, so we have RNA-Get as well, which is a product that is soon to be released. And this is sort of another sort of layer to it. So if you think of RNA-based analyses, it's usually a transcript abundance analysis. The main sort of data representation is a matrix where you have genomic features on one axis, and you have tissue samples, conditions, tumor types, what have you, on the other axis, and then the relative abundance of certain transcripts or certain genes according to sample type. So this API is essentially meant to allow researchers to dynamically slice and download subsets of large matrices according to their research interests. So some of these experiments are very vast, and these matrices are huge, potentially hundreds of thousands of columns by rows. So that might not be feasible for a researcher to download the whole thing in bulk. So the API allows you to specify, here I want these features, or here I want these samples. I'm only interested in a specific part of the experiment. So please don't give me everything. Please only give me what I want. Sorry, I forgot to mention that there are driver projects implementing each of these, and they're contributing to the standard development for each of these. So for RNA-Get, we have CanDig, Human Cell Atlas, as well as the Epigenome Consortium. I'll just go back to htsget. So we have Australian Genomics, the ENA, EGA, CanDig as well. Genomics England is actually doing a 100,000 genomes project, and they're going to be serving their alignment files over htsget, as well as the Human Cell Atlas is doing an implementation of this as well. For RefGet, again, it's Australian Genomics and the European Genome Phenome Archive. And there are other implementers as well, but I only listed the drivers. Okay, so those are 3 APIs. There's obviously many more for GA4GH, but I sort of wanted to focus on those just because they might be most relevant to like a cancer research sort of perspective. So you can sort of imagine that with the 3 APIs, intersecting the data you might get from the different APIs, you can conduct many different types of cancer research analyses. So if you look at RefGet and htsget, the intersection of that, having the reference sequence and having these BAM files might allow you to do somatic mutation calling, germline calling, copy number variance or structural analysis based on the, based on how the experiment was done that produced the BAM files, or that produced the htsget files. Something that would involve refget and RNA-get might be differential gene expression, or some sort of cancer epigenomics analysis, because the lessons learned from having these RNA matrices might have applications to epigenomics as well. And something that I thought that might be at the interface of all three is like gene fusion analysis. So what we're sort of talking about here is we're sort of a large scale or high concept idea we talk about at the GA4GH is this Internet of Genomics idea, where data doesn't necessarily have to be located in one place. But if you have interoperable APIs and standards, then a researcher might be able to access any data wherever it is in the world, regardless of how it's represented underlying. As long as the API that's presented in a certain way, that researcher should be able to access it and conduct analysis. So this is sort of a hypothetical example. But let's say we have a cancer researcher in Japan, and so they're the user, and then that document thing beside them is their GA4GH passport. It's another standard that I haven't quite gone into too much detail, but essentially it provides an authorization mechanism to different servers, essentially. And let's say they want to conduct some sort of cancer analysis, but they don't know the data that's available to them. So they might want to find data sets that might be applicable to them. So they might go to a search API infrastructure, and the search API is connected to many different systems that will give them information about what reference sequences are available, what HTS sequences are available, what RNA-get matrices are available. So they might be able to collate these many different data sets from different parts of the world and find something interesting to do with it. And maybe they want to conduct some sort of analysis. So what they can do is go to a terse API and collect some sort of analysis workflow, and then run it on a workflow execution service. So that's sort of a high-level thing, long-term, of what we want to do that involves many different GA4GH APIs interacting together. We sort of have something like this present day. It's sort of a more basic example, but it illustrates what is eventually possible with federated data sharing. So this is what we call the beacon. The beacon is a very basic API that allows a researcher to ask a question. So have you seen a mutation like this? And you provide a genomic locus with the reference allele and the alternate allele. And if you ask this to a beacon node, the beacon will respond with, yes, I have seen this mutation, or no, I haven't seen this mutation. And so what teams at the GA4GH have ended up developing is sort of a master node, which we call the beacon network. And the master node of the beacon network points to 76 beacon nodes around the world. And so when you ask the question, have you seen this mutation, to the beacon master, this request gets passed to everywhere in the network, and then you receive 76 answers about have you seen this mutation, yes or no. So this is federated data sharing. The data isn't located in one place, but because there is a common API standard, which is the beacon API, you can ask questions easily to many different places at once. Going from there, we have like a bunch of beacon nodes, but one of the challenges that we face with the GA4GH is, even if a node is saying that it correctly adopts the specification, how can we be sure that it is correct? So if someone says, oh, I'm a refget server, or I'm a htsget implementation, how do we know that the methods that they're implementing are correct and work according to the specification that we've written originally? So our answer to this, and our way of meeting this challenge, is to develop compliance suites that will run against our list of implementations that we track. They can sort of notify developers and implementers if their implementation doesn't meet some aspect of our specification. It basically gives us an insight into when a web service fails, or when it becomes unresponsive, or when it's not meeting the specification in some way. And again, we're not looking at underlying infrastructure, but it's just at the API level. So how this is done is pretty basic. It's not really too complicated of a concept. I'll sort of go through an example with using RNA-get. So we can assess compliance to the standard at various levels. So we can look at it at the HTTP level. So if we know we're going to ask a certain question, we know that we should get a certain response. So we can ask the question to RNA-get. We say, RNA-get expressions formats, and we're essentially asking the server to tell us all the file formats, all the expression matrices in what file formats do you support or do you have. And if the server responds saying 404 not found or 400 bad request, we know that this server is not meeting the expectation of the standard, because this is an endpoint that has to be implemented in order for it to be considered a valid RNA-get service. And then going forward from that, we can also look at the content that we receive that we download from the request. So given the matrix slicing example, if I ask for certain genes, I see those genes in the matrix. I should see certain tumor samples that I asked for in the matrix. I should see certain expected expression values when I compare against my golden standard reference data set. And we can also, perform more sophisticated requests to sort of interrogate the API. And only once it, passes all of our tests, we can say that yes, all the methods, however you've implemented it, are implemented correctly. So the way that we sort of do this now, we have different standards sort of implemented on a case-by-case basis, and it's not really a centralized approach necessarily. So a lot of my work has been focused on RNA-get compliance and ref-get compliance and moving into htsget compliance now. But essentially how we have it structured now is we just have like a compliance application that we schedule to build once every 24 hours. And during that build, we have a list of implementations that we're tracking, and we say, please run the compliance suite against these implementations and generate a report. This report will get pushed to somewhere on the internet, and it'll either tell us, yes, all of the implementations you're tracking have passed all scenarios, or it'll say, no, something went wrong with one or more implementations, and here's why. So this is sort of a good starting point for developing a compliance suite, and especially when the standard itself is being developed and the implementations and the standards have to be reworked consistently. So it's okay for now, but I think we want to long-term move to a sort of more comprehensive approach that can be a sort of centralized platform that will work with many GA4GH APIs at once. So something that we sort of have proposed is this idea of a generalizable GA4GH testbed, and this sort of comes out of the cloud workstream because they've so far been the most interested in having this general testbed model, but there are applications for the other work streams and the other APIs like the ones that I've mentioned. So this sort of infrastructure would essentially have our compliance suites available via a terse API from a sort of Docker container workflow repository like DocStore. These plugins would be reusable, so there could be a plugin for like refget version 1 compliance, htsget version 1 compliance, etc. And we can have some sort of way to configure and track the implementations that we want to track. And then the orchestrator service will run the plugin code against the implementation on some sort of schedule, generating reports on that schedule. And then these reports can be fed back into some sort of dashboarding and reporting service that we can monitor. Maybe we'll alert if we saw an implementation was working well, but for some reason it's failing for some reason. Maybe we can alert on that. Yeah, so this would be a more centralized approach to incorporate many GA4GH APIs into one sort of common platform. And going forward, if we develop any new standards or any new versions of existing standards, we would just need to develop a new plugin that would be available via a terse API. So something that we're going forward with is that we're not just, so compliance tests is sort of like the most fundamental level of how we can assess the quality of the implementations. But we're not just interested in doing compliance tests. We are interested in more sophisticated tests like benchmarking, so like performance testing, and also more sophisticated end-to-end processes that might, sophisticated test cases that might cover or touch many APIs in a single run. And this might simulate what a researcher's use case might be. So this sort of goes back to the Internet of Genomics concept I mentioned, but it would be what we want to eventually do is have these tests where we can run. So it might start with discovering a data set using a search API, and then we'll have some sort of test passport. So the passport will allow the testbed to authenticate to a certain set of GA4GH APIs. We can fetch data sets from a DIRS web service, which is another API for general genomic data. We could also authenticate to like htsget, refget, RNA-get, what have you. And then maybe we can also fetch an analysis workflow from DocStore via its tool registry service API. And then we can, now that we have the data set that we want to analyze and we have the analysis workflow itself, we can find some place to run that workflow like a WES API or a workflow execution service. So this is sort of integrating many GA4GH APIs and then essentially validating that they all work and that they're all interoperable with one another via a testbed sort of orchestration system. So how would the better compliance testing and having these interoperability testbeds and tests, how would that sort of aid our work at the GA4GH? Well, I guess it's pretty straightforward, but essentially we want to make sure that, not that we just have the standards for people to adopt, but that we are seeing that they are being taken up and that they are up to meeting the specifications. So if any sort of implementation doesn't meet the specification or many implementations don't meet some aspect of the specification, that might help us identify weaknesses or barriers to implementation. That might, pave the way for what should we address in the next version of some sort of standard, and it'll notify us when some web service goes online or fails to comply. Obviously, at the GA4GH, we don't want to say, this is the gold standard of htsget or something when it's not passing all the tests. So it'll essentially give us more insight into the landscape of how our standards are being implemented. So what's next in terms of this work? So we want to basically have a more standardized way of tracking our GA4GH implementations and deployments around the world. This would involve some sort of standardized metadata language. We want to develop a report schema that many plugins will be able to follow and to fit in with. And we also need to do the bulk of the development work for the testbed orchestration service and the reporting services. The existing compliance workflows that we have, we'll need to port over to this Dockerized model and make them accessible via TERS. And we'll need to design new workflows for other GA4GH APIs that don't currently have compliance. So that's pretty much it. I'd just like to close with a call to action. Maybe if you're a data provider and you're interested in making your data sets available via any sort of GA4GH API, please come contact me. If you're interested in contributing to standards work, you have ideas about how data should be made accessible over an API, please consider joining the calls for our standards. And yeah, I'd just like to close by saying thank you to everyone who is involved in this work. So obviously all of our work streams, implementations like DocStore, TERS API, and all of our refget, RNA-get, and htsget implementers. And that's pretty much it.